Hi guys, this is my second video from the Biology Goat. Today I'm going to do my second lesson on respiration. So we're going to go into a little bit more detail about what the energy released from the hydrolysis of ATP is actually used for, and then also look at the structure of mitochondria, which is where most of the reactions for aerobic respiration take place. And I'm also going to talk to you about something called the endosymbiotic theory, as well, which kind of makes sense to do in this lesson as we're doing the structure of mitochondria. So this is lesson 2. If you haven't seen my first lesson on respiration, please ensure you watch that first, just to make sure this one then makes sense. So this is going to be what is the energy released by and remember when we talked about in our last lesson breaking down ATP into ADP and PI, that reaction is called hydrolysis, and make sure you use that word because that's what they want you to say in the exam. So what is the energy released by the hydrolysis of ATP used for? And in reality there are loads and loads of uses of this energy, but I'm just going to give you a few that tend to be in most of the mark schemes that I've seen. Just a recap first of all. So we say that ATP is broken down into, make that line a bit longer, ADP and PI, and remember the I is just for inorganic because the phosphate group doesn't have any carbon in it, and we say that this is hydrolysis. I just want you to be aware that when this process happens, this hydrolysis reaction to split ATP into ADP and PI, we're also losing a phosphate group from ATP. So sometimes they also call it the dephosphorylation of ATP. So D means remove, phosphorylate is the phosphate group. However, I have noticed, particularly for OCR, that some of the mark schemes don't allow dephosphorylation if you're naming the type of reaction. So stick to this if they ask you what type of reaction it is. But I'm just going to write down that this includes what we call dephosphorylation because you're removing a phosphate group from the ATP. So it's D to remove, phosphorylation. This, because it's releasing energy when you're breaking this bond between the second and third phosphate group, you're releasing energy. This is also known as an exothermic reaction because you're releasing energy. So it's an exothermic reaction, which basically means release energy. In chemistry, endothermic is to take in energy or requiring energy. Exothermic is where we release energy. And that's the whole point of the hydrolysis of ATP, to release some energy so that the cells can then use it. So these are some examples of basically what this energy here is used for. So the first one is muscle contraction. Obviously some of these apply to animals, plants, all organisms basically, but some might only apply to animals. For example, the first one, muscle contraction. So muscle contraction is definitely one. And I'm going to give you 2 specific parts of muscle contraction that need energy. So the first one is breaking what we call the actin myosin cross bridges. At some point I will do loads of videos as well on muscle contraction, so this will all tie together nice and neatly, hopefully, if you ever have got the time or chance to go through all of my different videos. And then the other one is pumping calcium ions, which is Ca2+, back into sarcoplasmic reticulum after contraction. So I'm being quite specific, and these are the things that I would use if they asked you a question about what the what the energy is useful from the hydrolysis of ATP. So that's one of them. You can also talk about active transport. That's always a good one. If it was me though, especially if it was 2 marks, I would definitely say something like active transport, and then for the second mark give an example of active transport in organisms. So I'm going to just give you here a couple. So e.g. the sodium potassium, which I'm just going to write for short as that, sodium potassium pumps. You find these like in neurons, you find them in the like cells lining the PCT in the kidneys, which is needed for selective reabsorption. But that's a great example, the sodium potassium pumps. And I'll give you another one as well. So e.g. let's put the active transport. I feel like I should give you a plant one because it's all animals so far. The active transport of mineral ions from soil into root hair cells in plants. And you need to know which mineral ions plants need to get from the soil. So I'm just going to give you the examples that you need to know about now. So the mineral ions that plants take from the soil by active transport using the energy from the hydrolysis of ATP are NH4+, ammonium. So they need the nitrogen from this ion. They combine it with glucose to make amino acids. They can also take nitrogen in the form of NO3-, which is known as nitrate. And again, they combine the nitrogen to glucose or with glucose to make amino acids. They can also use the nitrogen to make nucleic acids as well. And then you've got also magnesium, which they need to make chlorophyll. And then you've got PO4-3-. They need this to make like the nucleic acids. They've got the phosphates in them, and obviously even ATP. If you think about, this is going to be needed to make ATP as well, which is a modified nucleotide. So there's some active transport ones. Also back to animals again, maintaining, I'm going to bring that line down from here. Maintaining body temperature, and one of the most sort of like significant uses of energy in an animal, like a mammal, a warm-blooded mammal, which we call an endotherm, a lot of the energy is used to maintain that like core body temperature. So for us it will be about 37 degrees Celsius. So the enzymes can work at that optimum temperature and things like that. So to maintain body temp, this is for endotherms, so like warm-blooded animals, which is actually one of the sort of like main disadvantages of being an endotherm. There's lots of advantages of being an endotherm, but actually you need a lot of energy to basically maintain that temperature. And this is so the enzymes can work at the optimum temperature. They can work really well. So we'll just put that on. So that's to maintain body temperature. I feel like all that can kind of go in there. Okay, also for what we call anabolic reactions, kind of run out of space. I'm hoping I can fit all of this on one page. So you've got anabolic, don't worry if you're confused, I'll explain this in a second. Anabolic reactions. So this is how I remember, there's 2 types of reaction, anabolic and catabolic. Anabolic is where you're adding molecules together to make a bigger molecule. So I remember the A for add. So if I just put A for add. So if you're going to join molecules together, you're going to make a bond. And whenever you make a bond, you need energy to make that bond. Okay, so I'm just going to put here. So I've got add. So energy needed to make bonds. So perfect examples are like peptide bonds if you're making a new protein, phosphodiester bonds if you're making like mRNA or replicating DNA, things like that. So e.g. peptide bonds and phosphodiester bonds. There's quite a lot, but I'm just going to stick to those 2, otherwise it'll just be too much. And I feel like if I give you just sort of like a few sort of main ones, that'll be definitely enough knowledge for the exam. And then I'm just going to give you a couple more as well. So we've got cell division. So looking at things like mitosis, meiosis, like the contraction of the spindle fibres when the chromosomes move to opposite poles, that requires energy, which is why cell division requires energy. So if I just put cell division, and then I'm just going to put here e.g. full contraction of like spindle fibres, which are actually microtubules. They're the same thing. Microtubules can be lots of things, but one of the things they always are spindle fibres, so tubules. And then the last one is things like movement of the cytoskeleton. So like you might have got a vesicle that's coming out of the RER that's got like a new protein in it. That vesicle will need to move to the Golgi so it can be like processed and modified. The movement of that vesicle requires contraction of the cytoskeleton because the vesicle will attach to the cytoskeleton, and literally contraction of that cytoskeleton will make the vesicle move, so it will move towards wherever it needs to go. So I'm going to do this as my last one, and I've kind of run out of space down here, so I'm just going to zoom back up there, and I'm just going to squeeze the last one in here. So let's put movement of cytoskeleton. And also I'm going to put this in with this one as well. If there's any process like endocytosis, exocytosis, or a flagellum moving, or cytokinesis, anything where the membrane is moving, also happens because of the movement or contraction of the cytoskeleton, and that always requires energy. So if you think about a neurotransmitter being released by basically exocytosis, that requires movement of the membrane and vesicles moving to the membrane actually, and both of those processes rely on a movement of the cytoskeleton or contraction of the cytoskeleton. So I'm just going to add in a few quick examples for this, and then we'll go into the structure of mitochondria. So e.g., so we've got vesicles moving. We've got like, I'm going to put endo and exocytosis. And I feel like that's enough, even if you've got a 6-mark question on what is the energy from the hydrolysis of ATP used for in organisms. That's quite a lot of stuff you could now talk about that the mark schemes will allow. Right, I'm going to just flip over my page, and what we're going to do now is go into the structure of a mitochondria in quite a lot of detail, because this is where a lot of the reactions in aerobic respiration take place. And I'm also going to talk about something called the endosymbiotic theory. So I'm going to call this the structure of a mitochondria. And I've updated my notes so many times over the last few years because I keep seeing new things in mark schemes, and I always think, oh, is that true? I never knew that. So I always end up Googling it, and then when I've confirmed that it is a thing, I add it to my notes. So first of all, mitochondria, they're about the same size as prokaryotic cells or bacteria. Okay, so about in size, they're about the same. So I'm going to put similar size to prokaryotic cells, just so you kind of like in your head have like when I draw this in a minute, you kind of know how big in real life it would be. So this is going to be a typical mitochondria. So it has an outer membrane. I'm going to draw it kind of like this. Sometimes mitochondria, by the way, they're not always like this shape. Like for example, I learned a few days ago that in sperm cells, the mitochondria are actually like arranged in a spiral around the sort of flagellum in the center, which is really cool. I'm not sure really how that's happened. I'm just guessing maybe that's the only way they can fit, because the sort of flagellum is so tiny, and that's where they need to be, because it's the flagellum that will need to contract so the sperm can swim to the egg. So maybe that's why. I'm not sure, but I just read that and I thought that was quite an interesting fact. So they're not always definitely this shape, but most of the time they are. So they have this outer membrane here. I'm going to add some notes as well to each of the sort of like labels. So we've got the outer membrane. Okay, and the outer membrane, sorry, it contains these protein channels, and they've got a unique name, which they're called pyruvate H+ syn porters. and we're going to talk about these quite a lot of detail like in a later lesson. So the outer membrane, it contains, I'm just going to move this down so you can see what I'm writing, and then we'll go back up in a sec, contains these things called pyruvate, H+, symporters, which are like specialised protein channels. And what they do, they transport pyruvate, hence the name, from the cytoplasm out here to the inside of the mitochondria. And this is important in respiration because the reaction that makes pyruvate is in the cytoplasm, which is out here, but then we have to get the pyruvate into here, which is called the matrix of the mitochondria. So it kind of makes sense that the outer membrane has these in it. A symporter is a type of co-transport protein that brings two molecules together in the same direction. So in this example, they'll bring pyruvate and hydrogen ions or protons in the same direction through the membrane. Antiporters are co-transporters that transport two things in opposite directions. So for example, a sodium-potassium pump, which does sodium in one direction and potassium in the other, they're going to be antiporters. So these are just the type of co-transporters that bring two things in the same direction. So they're going to be kind of like in the membrane here. So what I'm going to do, I'm just going to draw a few here, or just one there. So you've got there a pyruvate H+ symporter. So there you go. So there's your protein channel. Right, there's also an inner membrane. And the inner membrane on mitochondria has got these like projections or folds like this, and they're kind of like found all the way through the membrane. And they're going to increase the surface area because kind of one of the really important reactions that produces ATP happens on the inner membrane. So the more inner membrane surface you've got, the more ATP you can basically produce. So I'm just going to draw that like that. I'm going to make that inner membrane as well go through that little pyruvate transporter, because actually I should have, I'm just going to finish that off really. I could have left a space on the bottom, but these symporters, they actually go through both of the membranes, but I'll write that in a second. So you've got the inner membrane here, so inner membrane, and this has basically got folds that are called cristae, or crista is the singular, so it's got folds, called, so crist, okay, like that is the plural. Or if you're talking about just one on its own, you say the crista, or crista is like singular. And they basically increase, so up arrow just means increase, SA, surface area, for basically what we call oxidative phosphorylation. Don't panic about this, we'll go through this later. Increases the amount of ATP made. You've just got more surface for the reactions that are basically called oxidative phosphorylation, and that means you can make more ATP just by having this like extra surface, because the reactions can happen all around these folds as well as on the sort of main bits here as well. And also these, like I said, the pyruvate H+ symporter actually like goes through the inner membrane as well. So I'm just going to put as well from that, if I just bring that to there to show that the inner membrane's also got the pyruvate H+ symporters in it. Right, so that's the inner membrane. Then inside here, the filling, as it were, is known as the matrix in mitochondria. So this is the matrix, which is like just the filling on the inside. You'll also find in mitochondria either one or more loops of DNA, like you find in prokaryotic cells, okay? So sometimes it's just one, but sometimes there can be several. So I'm just going to draw two in mine just to show that. And then I'm just going to put a label up here. So one or more loops of DNA. And they'll join end to end just like they do in prokaryotic cells. So one or more loops of DNA. And these contain the genes that are needed to make the enzymes needed for respiration. So I'm just going to put contain genes that code for enzymes needed for respiration. Okay, they also have, and I only learnt this the other day, this isn't always true, just like it is with prokaryotic cells, but sometimes mitochondria can also have plasmids as well, so like extra loops of DNA. Sometimes they have none, sorry, that's supposed to be a circle. Sometimes they have none, sometimes they have one, sometimes they have several, but that's a similarity to prokaryotic cells. So I'm going to put may, because they don't always have them, may have plasmids, which is quite cool. Then you also need to know that they have their own ribosomes, but the ribosomes, they're actually smaller than the ribosomes you find in eukaryotic cells. We call them 70S ribosomes, whereas in a normal eukaryotic cell, which is a cell with a nucleus, these obviously don't have a nucleus, these are smaller, they're called 70S, they're 80S if it's a eukaryotic cell, so these are slightly smaller. So they've got these little ribosomes, and these ribosomes will be where these enzymes needed for respiration are actually produced. So these are 70S small ribosomes. So in eukaryotes, the 80S, they're slightly bigger. So just remember that in mitochondria, they're smaller. I also want to tell you a couple more things, and we'll go into the endosymbiotic theory. The space between the outer and inner membrane is called the intermembrane space, and you need to know that. So this is the intermembrane space there. And I feel like that's probably enough for the structure of mitochondria. What I'm going to tell you though now very quickly is where each stage of respiration takes place. So there are 4 stages of respiration. I'm going to write these down here, then we're going to add to our diagram where these are taking place. So this is true for aerobic respiration. So there are 4 stages of aerobic respiration. One of them, the first stage, is also used in anaerobic, but for now I'm just going to stick with aerobic. So you've got the first stage, which is glycolysis. Now glycolysis happens in the cytoplasm of the cell. So if I go back to my drawing, imagine this mitochondria is in a eukaryotic cell. It wouldn't be in a prokaryotic cell because obviously it's not going to fit because it's the same size as a prokaryotic cell. But also one of the things about prokaryotic cells is they don't have membrane-bound organelles. This is a membrane-bound organelle because it has its own membrane. So basically the cytoplasm in eukaryotic cells is going to be around here somewhere. So I'm just going to put here glycolysis. So this is where glycolysis happens, and I'm going to put a bubble around that so it's kind of like distinct from the rest of the notes. Okay, the second stage is called the link reaction. So we've got the link reaction. This is actually a very quick stage of respiration. And then you've got something as well called the Krebs cycle. It's got other names, but I'll tell you about that when we do the Krebs cycle video. So the Krebs cycle. These 2 stages both happen in the matrix of the mitochondria. So these are in the matrix. So just so you can kind of clearly visualize it, up here, matrix, so we've got the link reaction, and you've got Krebs is happening here as well. And then the last stage, stage 4, is called oxidative phosphorylation, which I've already kind of written here. So this is oxidative phosphorylation, and this happens on the inner membrane and in the intermembrane space. So this is inner membrane and intermembrane space. So again, I'm just going to go up and label this on the mitochondria. So I'm going to write it here actually. Imagine this bit of the membrane, so it'll be happening on this bit but also within this inner membrane here. So this is going to be like oxidative phosphorylation. So just to clarify, it's in the intermembrane space here and on the inner membrane all around here. I could carry on and keep going, but I'm not going to. I feel like that's enough. So that's the structure of a mitochondria. There are all the things you need to know about mitochondria. Now, I keep talking about this thing called the endosymbiotic theory, which I've noticed more and more is coming up in exam questions. And a lot of students don't seem to know what it is, because I don't feel like it's like specifically part of any of the specification points. So it's just sometimes missed or like not covered in textbooks or in lessons and things like that. So what the endosymbiotic theory is where we think that mitochondria and chloroplasts as well, actually, which are a very similar size to mitochondria, and they've got a very similar structure. We think they used to be prokaryotic cells or bacteria, and then basically one day, like thousands, millions of years ago, whenever, and a eukaryotic cell engulfed a prokaryotic cell, planning on basically digesting it and killing it, as normally happens with like bacteria, but instead realised that the prokaryotic cell was very good at doing something. So in this situation, it was very good at releasing energy, and the cell thought, actually, you're quite useful. Instead of digesting you, I'm going to keep you. And we think that mitochondria literally are what used to be prokaryotic cells. The same for chloroplasts. We think chloroplasts are what were prokaryotic cells. So the questions in the exam seem to be, can you outline the evidence that supports that theory? So what I'm going to do, I'm going to quickly explain it now while we've got this picture in front, and then I'm going to just write down a few bullet points of things you could write in the exam. So the first similarity between this and a prokaryotic cell is the size. The size is very similar. Okay, so this is roughly how big a bacterial cell is or a prokaryotic cell is. And that's also a useful way of remembering prokaryotic cells don't have mitochondria, they don't have chloroplasts, they're not going to fit because they're the same size anyway. So that's always a piece of evidence. Another is the 70S ribosomes. They're in mitochondria, they're in chloroplasts, and they're in prokaryotic cells, whereas eukaryotic cells have the 80S ribosomes instead. The circular loop of DNA is present in prokaryotic cells as well. Obviously, the mitochondria, chloroplasts as well actually can have several, but definitely this is a piece of evidence. So are the plasmids, another piece of evidence. The double membrane. Now be careful here, because prokaryotic cells have a double membrane, but the outer membrane is their cell wall, which is a peptidoglycan or murin cell wall. I wouldn't say cell wall for mitochondria and chloroplasts. This is called an outer membrane, not a cell wall, but you can say both have a double membrane or a double outer layer or something like that, and that seems to be sufficient. So what I'm going to do on my last page, and then we'll stop this video, is just talk about the evidence. So this is basically the evidence of and it's called the endosymbiotic theory, which basically is that we think, and again, this is a theory, so it hasn't been proven, and at some point, maybe in 100 years, they'll find actually it's a load of rubbish, but at the moment, the evidence that we've got that supports this theory is all the stuff we just talked about. So basically, the evidence of the endosymbiotic theory, which basically we think mitochondria and chloroplasts were prokaryotic cells, which are basically bacteria that were at some stage engulfed or taken in by eukaryotic cells. And the evidence that supports this is what you need to write in the exam. So the evidence, question mark. So it's normally about 3 marks. I don't normally see this much more than 3 marks. So I'm going to give you like definitely enough to get you 3 marks. So let's put all have. So we're talking about the mitochondria, the chloroplasts and prokaryotic cells. So all are similar size. I'll put sized. I feel like that makes more sense. So all are similar sized. We've got all have like circular loop in brackets loops, because obviously it can be plural for mitochondria, actually for chloroplasts as well, of DNA. I'm going to put all, now this, you've got to be careful here, may have plasmids, because they're not always there. Okay, all have the 70S. Sometimes it allows small in the mark scheme. To be honest, I don't feel like it's hard to remember 70S, so I would just say that. But if you do forget in your exam and you can't remember the number, just say small ribosomes. All have a double, like, outer membrane. And I feel like I've given you 5 things there. I feel like that would be enough to talk about the evidence of the endosymbiotic theory. If you start like really looking into this, there is other evidence as well. One's about these things called histone proteins, but I feel like that's a little bit harder to talk about because it's not always definitely true. I'll talk about histone proteins in another video. For example, there's 2 types of prokaryotic cell. You've got kind of your true bacteria or eubacteria, and then the ancient bacteria that are the archaea. Archaea do have histone proteins, whereas the sort of like true bacteria don't. So that's kind of a more vague, it's not always like really a thing. These are things that are always definitely a thing, are definitely similarities. Guys, I hope you found that useful. I've given you a lot of information there. My next video is going to be on sort of like the sort of things that are needed for respiration to happen, and then we'll start the first stage, which is glycolysis, and we'll go through that stage as well. Okay, I'll see you later. Have a good day.